I've been working on the aging of collagen, and we recently had a couple of publications out on the aging of tendon and how tendon functions. So this presentation is briefly going to go through what is tendon aging, because it's an obvious question and seems to have some obvious answers, but actually when you start to look at it, it's not so obvious. And then what are the chemical changes seen when collagen ages? Then I'm going to try and describe how collagen actually works in tendon. How does tendon chemistry change? Until recently, it wasn't really thought to change at all when you stretch it. But what we've been able to show is that chemistry is dynamically changing all the time. And so the changes that you get in the chemistry with age impact how the chemistry of tendon works. And then I'm going to try and bring it all together into some functional pictures, almost, well, they are cartoons, but I think this is very chemistry heavy, and I think people sometimes find it hard to translate the chemistry into what it actually means in terms of something they can actually see. So what is the aging of tendon? Typically in the literature, you find it described as an increase in stiffness or the Young's modulus, but equally you can find examples where there are where tendon stiffness has been shown to decrease with age. Generally, people believe that you see an increase of stress at breaking point, so the force required to actually break a tendon is believed to be higher in older tendons. Very rarely do you actually see real non-selective data in the literature. So the first thing we did when we started this project was to actually look and see what you can see when you stretch a piece of tendon, to see what to expect, and it was a bit of a surprise to us. So the work that I'm going to show here is all in black 6 mice. They don't develop diabetes with age, and you can broadly, for the purposes of this talk, say that 1 week of black 6 mouse life is equivalent to 1 year of human life. So a 20-week-old mouse is about 20 years old in human terms. So you can think of it as a young, mature adult. So this is what we get when we stretch tendon to breaking point at different ages. So on the y-axis, we've got the force required to actually break the tendon, or the force applied to the tendon. And along the x-axis, we've got the increase in length of the tendon, or the strain. So as you can see in young mice, there's a very homogeneous in this portion of this stretch curve, and then it plateaus. In older mice, mature, this early region is broader, and you've still got quite a bit of plateau. As you progress in mice through the age of the mice, the plateau region decreases, the amount of plateau that's there. And quite often you lose it altogether, and there's a tendency for the tendons to break. Now the normal operating percentage when you stretch a tendon for normal activity is in this region, this early region up to about 4%. So what you've got here is an elastic region, and here you've got what we call a plastic region. In this region, there's actually deformation of the tendon fibers going on. Sorry, there are lots of terms here that I'm using. I'll try and answer them at the end if anybody needs me to go over them again. So if you overlay those traces, you can see that the young mice show a much lower stress strain curve than the older mice. But really, once the mice have matured, it's very difficult to say that there is an increase in the Young's modulus, which is the steepness of this curve. These tendons have all been taken to be approximately the same size, and this is one reason why it's actually quite difficult to say that tendon actually stiffens with age. Because when you actually look at the stiffness of tendon with the diameter of the tendon and with age, so here we've got in orange the young tendons, and in blue here we've got very old tendons. There's a difference in the way the tendons mature. So if I were to look at tendons in this sort of region, it would appear that young tendons were very much stiffer than old tendons. But if I were to look in this region, it would appear that young tendons are much less stiff than old tendons. So you've got to be very careful, and the graphs I've shown you are all taken in this sort of region of tendon diameter. So saying that the tendon is stiffer with age is not really a good measure of aging. There is often an increase in the breaking stress, but equally there's quite often a decrease in the stress required to break a tendon in older mice. What we really are talking about is a decreased ability to undergo a plastic change, so to undergo a deformation without breaking with age. So that reflects the ability of the tendon to absorb the stress that's being applied to it. And that's linked into the fragility of the tendon. If it can't absorb the stress and it can't deal with the stretching process, then it's actually more likely to rupture. And so in the older animals, you tended to see the tendons rupture much earlier. And you also see this in diabetic mice. So chemists control the physical properties of polymers by cross-linking, introducing cross-linking, and fine-tuning polymers with the degree of cross-linking that they introduce. And biological systems do the same with collagen. So I'll quickly go through some chemistry here. There are cross-links in collagen that are derived enzymatically through lysine oxidation to generate an aldehyde, as shown here. And this can then go on to form an imine with an adjacent lysine or hydroxylysine within the collagen structure. This is a reversible reaction, and there are a range of molecules that can be formed that are based on this chemistry and can be considered reversible. Over time, some of these will mature into structures like this, which are considered to be irreversible. Then there are non-enzymatic processes going on. You'll all have heard, I'm sure, of advanced glycation end products. These are formed through the reaction of sugars with amine type residues within the structure of the collagen. And quite a few structures have been found, but in our studies, if we can't find a structure in 0.4 of a milligram of material injected onto the mass spectrometer, then we consider that it is unlikely to be impacting the physical properties of the tendon. And so in our studies in mice, we tend to only find pentosodine at a level we consider significant and glucosepane. And these are all irreversible structures. So if you actually go on and look at the development of these irreversible mature crosslinks with age, you see that they do indeed increase with time, as you might expect if you were to say arbitrarily that tendon stiffens with age. And this would be what's expected. And this is actually where most people, most authors have stopped. They said, haha, these irreversible crosslinks increase with age, therefore they're causing the issues with age. that we see in the physical properties of tendon. But actually, if you look at the reversible crosslinks, and the main reversible crosslink in tendon is HLNL. In the early phase, up to maturity, there's a very rapid drop, but there's still a very, very significant drop in this crosslink with age between young adulthood and basically the point at which the mice are about to die. So if you sum all the irreversible crosslinks, and I'm sorry, this is a bit of a busy slide, and compare that to the decrease in adulthood here, then in green at the box at the bottom, it's got summary. There's essentially an increase in irreversible crosslinks of 1 for every 5 collagen molecules, and a decrease in the reversible crosslinks of 4 in every 5 collagen molecules. So there's a net decrease in crosslinking of 3 crosslinks for every 5 collagen molecules. So a statement along the lines of collagen stiffens because there's an increase in crosslinking is clearly not correct, because there's actually an overall decrease in crosslinking. So what on earth is going on? Well, one of the major changes that we see is the glycation, so that's the addition of glucose to the side chains of lysine and hydroxylysine. And in green here, we can actually see the addition of glucose to lysine. So the question is, what is this doing? How on earth can the addition of glucose to lysine be making much difference? Does it make a difference? Is this what's causing the changes in properties that we see, or contributing to them? So we then did some work with diabetic mice, which I'm not going to show you here because I don't think there's time, which basically confirmed that glycation of lysine was important in the properties, but we didn't have any mechanistic proof of what might be going on, or the involvement of this glycated lysine. And what I will go on to show in a minute is actually that the glycation of lysine is involved in the stretching process. So to understand the changes that I've just shown you, you have to understand a bit of the chemistry. So basically, the oxidized lysine and hydroxylysine can react together and form an imine in the structure, but this is a dynamic reversible process. You could also imagine this as glucose reacting with the lysine and forming an imine. The chemistry is exactly the same, and one of the things about this is that all the chemistry of the irreversible crosslinks and glycated lysine are linked in very complex equilibria. Now, fortunately, you can freeze the equilibrium by actually adding reducing agents, which basically reduces out this bond and reduces out the aldehydes. So you can actually look at what happens at a point in time. So if we look at a simple stress strain profile where we've stretched tendon to breaking point. Now this is tendon, all of the same age, but in orange we've got ones where we haven't fixed the reversible chemistry, and in black we have fixed the reversible chemistry. So the impact in this form of testing of stopping the reversibility of the chemistry is to actually remove this second phase here, this plateau phase. And it causes the tendons to just keep increasing linearly along here until they break. So clearly the reversible chemistry is important in this phase. Now, as I said earlier, tendons generally work in this phase unless you overstretch them. So what could be happening in this phase? And to see this, you have to do a slightly different form of testing. So what we're doing here is we are taking a tendon and we are stretching it up to 4% and then allowing it to relax, and then stretching it again, and then allowing it to relax. And we've displayed 4 cycles of this stretching process, where we're only showing the stretching. We're not showing the relaxing period. That complicates the profile and makes it quite hard to see what's going on. But what we've got here in the tendon that's not reduced is, after the first cycle, there's a big change, and then subsequent cycles are all very reproducible. And this is actually an elastic profile. So the tendon returns back to its original position and then is following a very similar process each time. Now, something to bear in mind is that in the literature, people quite often condition tendon. So they actually stretch it several times beforehand and remove this first cycle. I'm going to show you that this first cycle is actually important later on. Now, if you stop the reversible chemistry from going on by reducing it, fixing it, what you get here is a series of cycles, and each time the tendon starts in a different position when you start to increase the force that you apply to it. And what we're saying here is that the tendon is actually getting longer each time you stretch it. So it's actually being deformed on each cycle of stretching. So what we can say here is that the reversible chemistry is important in somehow coping with this deformation and retaining the elastic properties of the tendon. So the important box here to look at is the green one, which is the glycation here of lysine. Now, this dotted line shows you the level of glycation before you stretch the tendon. And what we've done here is we've stretched tendon, and then we've looked at the difference between the stretched tendon and the unstretched tendon, while the stretched tendon is still under tension. So we've frozen the chemistry at the point where it is stretched. And that's really important because this chemistry is actually very fast. And if you don't do that, you miss what you're seeing, certainly with these other things going on here. The glycation, I'll just say, the exception is the glycation of lysine. The glycation of lysine is not reversible. What we're saying here is that there has been a drop when you stretch to 4% of the amount of glycation on the collagen. So you stretch the tendon and you lose some glycation in that early phase. And if you increase the stretching up to breaking point, you don't really increase the amount of glycation that you lose. So what we're saying here is that from this first stretch, we're losing the glycation, so we're losing glucose from the lysine residues on the tendon, and that then allows this elastic behavior to occur. So what we're saying, this shift here is a stiffening. So what we're saying here is the difference between this and this is saying that the first cycle is stiffer tendon than the second cycle. So that is some mechanistic evidence that the glycation of lysine, the addition of glucose to a lysine residue, actually causes a stiffening of the tendon. So what about the other changes that are going on? So the big change, although it looks small here in comparison to the others, is because the scale on the others is different. The big change, and this is the major crosslink in tendon, is here. And actually, when you stretch tendon to 4% and look at what's happening in the stretched tendon, there's actually an increase in the crosslinking of the stretched tendon. You then keep on stretching it to 10%, so this is in the plateau phase of the stress-strain curve, you actually see a decrease in the cross-linking. And again, when you break it, you've lost some cross-linking. So this might look like quite a small change. So what I'm going to do is now try and show pictorially what we're talking about in terms of the number of bonds. So what we've got here in blue are collagen molecules. And what I've shown in green and red are the HLNL crosslinks, as I was just talking about. So these are the reversible crosslinks. If you draw them in, and you draw in the number that you would expect to see in 30 collagen molecules, this is the sort of picture that you get. And you can see there's actually a hole in the collagen structure here. And if you were to extend this structure, there would also be some other holes in the structure as well, which are hard to see because I've taken one unit of 30 molecules. So if you were to decrease by 0.1 mole per mole of collagen, you actually remove 3 bonds. And as you can see here, there's the potential for a fracture line to develop within the polymer. And so this is essentially a weakening of the tendon structure. Equally, if you were to increase the amount of cross-linking by 0.1 mole per mole of collagen, it's an increase of 3 bonds. And what you see here is we can potentially fill the gap in the collagen structure that I highlighted before, and you can start to form cross-links between other planes of collagen in this model. And tendon is being stretched, so what we've got here is a piece of tendon as before, and we're pulling it at both ends, causing stress to be developing within these bonds. The act of pulling increases the rate of hydrolysis as these bonds are stretched, and they break. But then when they've broken, new bonds can start to form with adjacent collagen molecules. And this is a mechanism for actually lowering the stress within the tendon structure, so the breaking and reforming of bonds. So finally, just to show you the difference that we're talking about between old and young tissue, here we've got the reversible crosslinks in young tendon. We've got the reversible crosslinks in old tendon. As you can see, there are many fewer and so many more gaps in the structure. If we then add the equivalent number of non-reversible crosslinks that old tendon has, you can see that the picture looks a bit better. There are not so many holes, but there are still quite a few holes. So this would lead to increased prevalence of breaking. The structure is overall a weaker structure. If we were to look at diabetic tissue, what we see is in young diabetic tissue, the level of the reversible cross-linking that we're talking about here is much, much lower than in normal young tissue. The difference is between this structure and this structure is that in this structure we've got much more of a crosslink called HHMD, which is a different sort of reversible crosslinker. So the overall cross-linking of young diabetic tissue is similar to the old non-diabetic tissue, but the type of cross-linking is different, and that leads to the differences in behavior between diabetic tissue and old tissue. Now, very quickly, because this is what we all want to know, how to deal with these problems, what would we do? Well, at top of my list would be to decrease the level of lysine glycation. This should improve the amount of the extension of this elastic phase, and it should prevent the premature breaking of tendon fibers. Secondly, what I would do is, step back, got myself confused. I would decrease the level of glycation because it would increase the elasticity in this region. So I'm very sorry if I've confused anybody. I confused myself there. So decrease the level of glycation, you will improve the elasticity in this functional region. If you increase the level of reversible crosslinkers, you will actually increase the amount of plastic phase that you get in these fibers, and essentially you'll make more of these look like these three here, which must be good. You would get fewer breaking quite so early. Then there's one which is probably quite controversial, or at least my view is, I'm really not sure about the idea of decreasing the irreversible cross-linking. If you were to do it, I would think that you would have to have increased the reversible cross-linking. If you start to remove some of those irreversible cross-links, my prediction would be that you get more of these tendons breaking earlier. So you need to have more reversible cross-links in there to prevent that from happening before you decrease the irreversible cross-links. Now, I'm sure there'll be people who won't agree with me, but you know, this is just my view in a minute. So here are the acknowledgements. Melanie and I did most of the experimental work on this, but we are helped with Arena and Isabella. All the stats, and the stats is really important in this sort of work, was done by Anne. And these are the papers that I recently published that have more details if you would like. And this was funded by the SENS Research Foundation. And I really don't believe that I would have got funding myself through the research councils to do this. I feel there would have been a lot of resistance to the idea that tendons didn't stiffen because of an increase in cross-linking. I would expect them to have found that quite radical. And I'd also like to thank Aubrey, because if Aubrey hadn't asked me to a meeting sort of 7 or 8 years ago and said, you know, what do you think of glucosepane? What do you think of cross-linking in collagen? I wouldn't be working on this because it wasn't on my radar at the time. And so he's actually got me thinking about this and then supported me to actually see what goes on. So thank you all for listening.